Welcome to onAIRR, a podcast from the AIR community. The community organizes and coordinates researchers studying all aspects of B and T cell receptor repertoires. The podcast is supported by the Antibody Society. For more information, please go to antibodysociety.org. This podcast has a special focus on clinical use of the adaptive immune receptor repertoires. We will look at how repertoires are currently used in the clinic and also discuss different opportunities where repertoires can be a great addition, the reasons why we are just not quite there yet, and how to overcome the obstacles. We are happy that you joined us for this episode of onAIRR. Welcome to the 19th episode of onAIRR, the podcast of the AIR community with a special focus on clinical use of the adaptive immune receptor repertoires. Today, we are joined by three people. We have Lauren Williamson from Vanderbilt University Medical Center in the US. Hello, Lauren.
Hello.
We have Robert Carnahan also from Vanderbilt University Medical Center, also in the US. Welcome to the podcast, Robert.
Thank you.
And then we have Matthew Vogt from University of North Carolina. It's very nice to have you here, Matthew.
Thanks for having me.
The podcast is hosted by me. I am Ulrik Stervbo.
And me, Ching Ding. Hello, everyone, and welcome. Why don't we first start off with introducing sort of yourself, your background, and a little bit about how you got interested in immune repertoires. And we can go in the order as Ulrik introduced you guys.
All right. I'm Lauren Williamson. I'm a research assistant professor at Vanderbilt Center for Antibody Therapeutics. I did my undergraduate studies at Virginia Tech in biological sciences and biochemistry, focusing on microbiology and immunology. After undergraduate studies, I was a postbac at the NIH. And then I did my PhD at Vanderbilt University in the lab of James Crow, which is where I'm still at, even as a research assistant professor. I've always been interested in microbes and immunology, even based on my undergraduate studies. I just think it's very interesting how the human immune system responds to microbes and trying to better understand that, which is why I'm studying what I'm studying.
So I guess I'm next. I guess I have some similarities and differences to Lauren. I was an undergraduate at Indiana University focusing in microbiology as well, and worked for Eli Lilly for a couple years, and then came to Vanderbilt for graduate school thinking I would do infectious disease, but took a kind of a right turn into cell cycle and ultimately cancer biology, including a postdoctoral studies in cancer biology. But in that postdoctoral studies, I got interested in antibody therapies, mostly for cancer at the time, and kind of went a decade off into just doing antibody discovery and things like that, which really suits me because I really like the strategy of finding needles in a haystack, I guess. So that was something I really enjoyed. And then had an opportunity to join the Center for Antibody Therapeutics as a faculty member to sort of come full circle and bring the antibody stuff I had been doing for 10 years in cancer back to my first love, which was infectious diseases, and sort of put those two things together.
All right. So then me. So I am an assistant professor in the pediatric infectious diseases department and the microbiology and immunology department at UNC here in Chapel Hill. And but I came here by way of Vanderbilt. So I was also part of the CROW group, the now named Center for Antibody Therapeutics. Or is that what it is, guys?
Yeah.
Okay. Yeah.
VCAT.
Yeah. It was a different name when I was there. So anywho, I also kind of like started in the virology world. So even though my PhD was in immunology, I worked in a lab of a guy named Mike Diamond, who very much lives at that intersection of immunology and virology. Kind of have always been very fascinated with how viruses work, how they can be not true organisms, but also things that could totally like take down society, which, with our pandemic recently, I was one of the many people who had, personal statements on things I applied for saying, I think, viruses are interesting because like they could cause a pandemic. And that was, then a pandemic happened. Promise I wasn't part of planning that or anything like that. So that study ultimately led me to get pediatrics training. So I am also a clinician a couple weeks out of the year. So I was at Boston Children's Hospital for that. And then I came to Vanderbilt for my infectious diseases, like subspecialty training in pediatrics. And so that was part clinical at the beginning, but then I spent 3 years in the Crow Lab, and kind of, again, being surrounded by a group of people there who, think about immune repertoires, but also, in my case, really focusing on a particular set of, antiviral antibodies that I was interested in. And now have my own group. We're almost 5 years old here at UNC. And we now continue to search for human monoclonal antibodies that bind to enteroviruses of all varieties, actually picornaviruses of all varieties. So I have a very microbe-focused interest in the immune repertoires, but I'm very intrigued about immune repertoires from other perspectives too. It's just our work is very microbe-focused.
That's all very nice. So you're all set together by Vanderbilt University.
Yes. Yeah.
Yep.
But it's not the only thing that ties you together, is it? So there is this ReVAMPP consortium.
Yeah.
So you're all involved in this.
Yeah.
Can you, one of you, tell us a little bit about the consortium?
Well, probably all of us can tell you a little bit. I guess I'll start by saying, just to introduce what ReVAMPP is. So ReVAMPP is essentially the NIH's response to the pandemic, in the sense of not so much response to the particular pandemic we had, but in processing it and thinking about the lessons learned from the COVID pandemic. How could we think about what we did well and what we didn't do so well, and to prepare for the next pandemic? So that's kind of the premise. And the idea is that there are only a limited number of viral families that actually cause human disease. And for the most part, we know what those are. So we shouldn't be truly surprised when a pandemic occurs, because we more or less know where it will come from, though I think we can often be wrong. As I've said, I think maybe to other people, I was convinced, if someone asked me before the COVID one, I would have said it's flu will be the next pandemic. It's always flu. It always will be flu. That is completely wrong, but that was what I thought. Anyway, but we know the families, and the idea is we should start to think about how to address those families now. So maybe what some people do or don't know is the response to the COVID pandemic was greatly aided by people studying other coronaviruses, right? Barney Graham, Jason McClellan, Mark Dennison, these people who had really spent a lifetime studying some of these coronaviruses and basically figured out the game plan. How do you make a vaccine? What kinds of antibodies? What would the targets of the antibodies be? All this was kind of known for other coronaviruses, which is why we were so able to rapidly respond as a scientific community. So the lesson learned there is, think about these things ahead of time and do prototypes or related viruses. And so that's what we're all doing for vaccines and antibodies, using prototype viruses from every one of these viral families that cause disease, and think about, well, what would be the strategy if you were to try to make a therapeutic antibody to an arenavirus, right? Or fill in a picornavirus, as Matt and Lauren both mentioned. So that's kind of the idea. And these are diverse consortia across the United States. I believe there are 5 of them. We are all on one together. Lauren and I also participate in some other ReVAMPP activities, and maybe Matt does as well. I don't know. But there's one unifying one that 3 of us work on together.
Could you speak a bit more about sort of the structures? I'm very unfamiliar with, you're saying there's one unifying one, but there seems to be, I guess.
Right.
Yeah.
So each group put in what they wanted to work on. Matt, Lauren, I think each group had to pick 3 viral families. Is that right? They would work on
2.
2.
Yeah. Yeah, 2.
I don't want to hog all the. Yeah, go ahead. Somebody.
Yeah, I would say unifying, and the 3 of us are part of one program, ReVAMPP Consortium, but there are multiple other ReVAMPP programs that are, some of us are part of those as well, but other members, within the scientific community are involved in. So it's a pretty, nationwide effort to study prototype pathogens, which Rob alluded to, of trying to better understand correlates of protection, vaccines, antibodies, focusing on both sides of that. How is immunity responding to these prototype pathogens, and can we apply them broadly to the family? to where, if there is a pandemic of a related virus, we can apply those techniques. Even if we have therapeutic antibodies, can they be broadly reactive to something that might cause outbreaks?
So the consortiums are organized by the different families of viruses, but it seems like there's also overlap, or?
Yes.
Okay.
There's intentional overlap. They intentionally want different approaches. So each, like our consortium, the VUMC one, we're the prime site, VUMC, Vanderbilt. And so we have three viral families we're focused on, I guess. But some of those other consortia have other viral families. I also just want to I think it's a really important part of ReVAMPP that I think is a little different than some other NIH programs. We talked just now about the strategic goal. The strategic goal is to define a platform that's capable of responding to virtually any pandemic, okay? But there's also a translational and what I would call a practical goal. Many of these viral families are already causing epidemics and outbreaks, and literally in our monthly deliverable reports, we have to report, are we progressing towards actual medical countermeasure solutions for those specific viruses? And we're accountable for that along the way. So we're not just doing theoretical strategic work, we're using practical applications of technologies to develop therapeutics that we could put in the clinic right now in the next couple of years, while also extracting those lessons and strategies. So
I see. So the other consortiums are utilizing different other strategies than what you guys are doing. Okay.
You have three viruses that you work on, and the idea is to look at how they infect cells, how the immune response works, and how you can develop good vaccines against these three viruses. Is that like correct summary of what you do?
So I'll take a stab at this and say that I think the overall goal, as Rob put it, right, is pandemic preparedness. Like that's the overarching goal, with translational output along the way being antibodies and vaccines. But it's not just the funding for this grant is not just focused on, hey, Matt Vogt and Lauren Williamson, go find antibodies against coronaviruses. It's also, what are those immune correlates of protection? So like, what would a vaccine require to be able to be protective? And so we might focus on, just one individual virus, like poliovirus or enterovirus D68 or something like that. But then the goal is, if we define immune correlates of protection for one of those viruses, which might involve in vitro work in cells, like you said, it might involve making mouse models. In fact, we do all of those things as part of our overall consortium that we have put together. And then also, my group does, and Lauren does, we do actually look for monoclonals. And then if we find really good monoclonals, we say, well, one thing we might want to do is make sure those vaccines are capable of, stimulating antibodies that target the site that we find is like the really, really strong neutralizing site. So, it's trying to put it all together so that, as Rob said, we have this like overall pile of knowledge that if there's a new picornavirus that comes out of left field, or there's one of the picornaviruses that we know, but, it gains some mutation or it recombines with another corona or picorna or whatever, that we kind of have these, this whole toolkit to pull from. Because that really is, we got lucky with coronavirus being the pandemic, which is wild to say, but is so true because of all of that concentrated effort over decades. We had a playbook. And so we're trying to, I would say really the overarching goal is build a playbook. And so there's a lot of approaches that go into that.
So then I guess going back to your piece about defining the immune correlates of protection, is your guys' group within the ReVAMPP consortium, your platform is sort of identifying from patients or people who've been infected with a certain virus who've then have these neutralizing antibodies and then identifying those neutralizing epitopes to then design a vaccine. Yes, can you expand upon sort of what is this platform that you're talking about?
I'd say that is an approach we are taking among many approaches. And that gets back to that idea of we're trying to build a playbook with many different pieces of information. And so, I keep mentioning Lauren and I, it's because we're on the same sort of project within the overall consortium where we are indeed trying to get potently neutralizing human monoclonal antibodies against viruses like rhinovirus C, which can be especially severe in young children, also seems to have a role in causing asthma. Nvirus D68, which also is a very weird respiratory pathogen that can make people who don't have asthma wheeze as if they have asthma. It can also paralyze you, just like poliovirus can. Nvirus A71 is another one that can cause big problems as far as typically causes hand, foot, and mouth disease, but also can cause neurologic disease like encephalitis, can actually kind of make your heart and lungs stop working really well, and they just sort of flood with fluid, which is, as you might imagine, a terrible thing to have happen to you. So, those are those prototype pathogens that we've chosen because, again, even in the here and now, those are causing awful diseases in humans. And yet, as we identify, so we have identified, for example, for Nvirus D68, we pretty much know there's like an epitope. We have it very well defined at a molecular level. There's an epitope that is like the epitope you want to hit with a super potent neutralizing antibody. It cross-reacts to every Nvirus D68 isolate we've ever gotten our hands on, including stuff from the 60s, including stuff that circulated after we had pulled the B cells out of the people from which we isolated those antibodies. So they're sort of like backwards compatible and forwards compatible. So like that's finding the playbook. That's the good epitope, right? Simultaneously, we have a collaborator in Raul Andino at University of California, San Francisco, who's working on generating mRNA-based, right? I think it's mRNA-based vaccines for different coronaviruses. He's targeting those same prototype pathogens. And so if we have a good antibody, we can send him that antibody and say, hey, test this against your immunogen and make sure that that epitope is present in your immunogen. Or similarly, he can immunize mice and we can see if we can pull those same antibodies out of mice and make sure that those antibodies are getting generated or something like that. And so that's like a crosstalk between monoclonal discovery and vaccine discovery. And then in the meantime, how do we even evaluate that these things work? So we need a good Nvirus D68 model in which to test the Nvirus D68 antibodies or vaccines. So my lab has a mouse model of paralysis. And so we can, at least regarding paralysis, not so much regarding the whole asthma-like respiratory disease, but at least regarding paralysis, we have some ability to test for that. So, that's pulling together many threads. We also have structural, I guess I would call them structural virologists in our group who, can do things like take those individual monoclonals, make Fab fragments, mix them together with purified virions and see get that really true molecular level detail of what is that epitope. But also we're working on things with who is it, Robert Kirchhofer at University of Wisconsin in Madison, Wisconsin. He is doing something similar, but with polyclonal antibodies. It's called MPEM. Do either of you guys, Robert, Lauren, do you remember what the MPEM acronym stands for? Off the top of my head, I'm blanking. It's like electron microscopy, polyclonal EM. Electron microscopy or something.
I don't know. Polyclonal.
Epitope mapping,
yes.
Polyclonal something.
Electron microscopy.
Yeah. So there we go. So some order of those EMs and PEMs. But yeah, so the idea is he's, looking from, so another collaborator, Jim Gurn at University of Wisconsin, has this like cohort of children he's been following in some cases for 20 plus years of their lives. And at various points, he's sampled blood from them. He's taken swabs of their noses. He has a really detailed map for these individual people of when they got particular rhinovirus infections, and then samples from before and after particular rhinovirus infections. And so we're taking the polyclonal antibodies from those older, the pre-infection samples and the post-infection samples, when we know after the infection, they've generated a really polyclonal serum neutralizing response. And so then we're going to try to do this polyclonal epitope mapping. We take Fab fragments from all of those purified polyclonal antibodies, basically mix them with these viruses, and then you just can sort of see all the different densities that, like the different, I guess I would call them like families of densities that form around these virions. And you can start to identify what are the sort of major epitopes. And it may not always be at a completely molecular, like this atom hydrogen bonding to this atom level, but it gives you a footprint. And so again, you kind of now have an idea that, well, we know humans can certainly make antibodies against those epitopes. If there's something that's different in that post versus pre, then you really got an idea that, all right, we know this guy makes, this particular human made a potently neutralizing response, and they make antibodies against these epitopes that they weren't making before. Okay, so now we're sort of like bootstrapping. And then if Lauren gets a monoclonal that also neutralizes really well, and then we can say, oh, look, it also seems to hit the same spot. Now we've kind of got an idea that humans are capable of making antibodies against that spot in the big picture sense. This is the one to go for. So make sure your vaccine immunogens elicit antibodies against those. Also, if we can make a human monoclonal in the future because there's a rhinovirus C outbreak or something, we want to make sure we are looking for antibodies. If there's a way to bias towards antibodies in that epitope, that's a way to try to do that as well as a sort of response platform for pandemics.
Yeah, I think that explains things pretty well for coronaviruses. I think there's at least in the VMC ReVAMPP as well as others, there's always the vaccine and antibody component, and having them work together. That is kind of the point, and it's not two separate things, but sending things back and forth, validating immunogens, or making sure antibodies are can be therapeutics as well. So it's just kind of having that network work together is also kind of part of the whole effort.
Just maybe say something that may or may not be obvious to the listeners is to respond to a pandemic. I think, in our opinion, you require both vaccines and therapeutic antibodies. So vaccines are a scalable, easily scalable, sort of hugely distributable technology that can allow people to develop their own sort of immunity to the pathogen, but not everyone can develop that immunity. Vaccines don't work for everyone, right? So we need therapeutic antibodies for those who can't. Also, vaccines only work when given prophylactically. So only ahead of time. You have to have the vaccine not only, and it can't just be minutes before, it has to be, depends on the vaccine, it can be days, weeks, months of time to get immunity. Whereas therapeutic antibodies, as a general rule, can be given at the moment you're exposed, or even in some cases, if the antibody and the dosing has been worked out, post-infection, even like in the hospital when you're severely ill, antibodies can still provide benefit, whereas vaccines have no benefit in those situations.
So your work is like twofold. So you want to identify good neutralizing antibodies as a benchmark for vaccines, but you also want to identify antibodies that can be used therapeutically also. But I guess then those will have two different properties, right? Because I imagine with a therapeutic antibody, you would like to have one that's cross-reactive in a sense, so it captures so many mutations as possible, because you need to produce a lot before there's ever a pandemic.
It's really, I won't take a whole long, that's a very complicated question. It depends on the pathogen. Yeah. Some pathogens actually at the key neutralizing epitopes actually don't change very much, right? Just we can go into many reasons why that is, whether they're RNA versus DNA viruses and how they circulate and how they get transmitted and a million other things. So some viruses you really don't need to focus so much on. Like we're all programmed to think about that now because COVID threw us such a curve ball in a way of being so changeable. Many viruses, we don't worry about that. And we have strategies for even the ones that change a little bit where we could, for example, do a 2 antibody combination. So even if there's a little bit of change, we can accommodate that. So in a sense, are the antibodies that we would provide to vaccine people and the ones that we use for therapeutics different? It depends. I don't think that we could say one way or another. They could be the same set. They could be distinct depending on the pathogen and the goals.
Yeah, and I think getting at the breadth aspect, it's most of the time neutralizing antibodies are virus specific. It's not always the case, and it does depend on the pathogen, but usually you do take a hit with neutralizing activity when you have breadth. So that is a component if you want something that's broad to the whole family or specifically to what is that viral pathogen or other pathogens. That comes down to diagnostics as well, of figuring out what the person actually has. But if you want something that you don't have any idea and you're like, well, but the virus family, that's where breadth comes into play. to give you something at least to hopefully give some benefit.
I think I'm going to bring up a dirty word, at least in Vanderbilt eyes, but there are also, so when we think of neutralization, typically we're thinking of a mechanism where antibody opsonizes virus and that virus is inert, either because the virus can no longer do what it needs to do to like release its genome. So it's just like it's trapped in a particular conformation and that it needs to change and it can't change anymore. You can also do things like block binding to receptors. So the virus just like can never get into the cell to, keep going. Those are two of the more common, like potently neutralizing antibody mechanisms, I would say. But there is this phenomenon of non-neutralizing antibodies where you can opsonize a virion just fine. Like they stick to the antibody, the antibodies stick to the virus really well. The virus is still actually capable of infecting cells. So you don't block receptor binding, you don't make the virus inert. But by opsonizing virions, you sort of prime the immune response via what we call Fc effector functions. There's this whole field, I would say now, that has built around this study of Fc effector functions of antibodies. And so that's where you might have a good breadth antibody that might still provide some benefit via the Fc effector functions. That said, like ideally, you'd want all of it, right? You'd want to have something that is both broad and potently neutralizing. And then like the Fc effector function is just sort of a bonus if you can get it. But there's, I don't think these have to be exclusive of each other, much like I don't think vaccines eliminate the need for antibodies and monoclonal antibodies eliminate the need for vaccines. That doesn't happen. I think this is also part of why, getting it back to the whole ReVAMPP strategy, they're trying to have many different groups work on some of the same virus families because, someone might be a little more interested in finding these broadly reactive antibodies that may not be neutralizing, but may, at least when given prophylactically, still give you enough of a leg up on the virus that it's not going to kill you. It may infect you, but it may not kill you, which I think we can agree is a good thing. Yeah. And so, and then maybe, but we're more focused on just that potently neutralizing thing. And if we at least know what that epitope is on one virus, at least for picornaviruses, to keep bringing it back to that, since that's what I think about a lot, they're all shaped pretty similar to each other. They have their differences and whatever, but overall, it's a pretty similarly shaped virus. Pick one picornavirus, pick another. The themes are the same. And so if you can try to identify a spot that, the structural genes all come together and this antibody sort of hangs across the structural genes. This is a very specific example of an antibody we have. It seems like it's doing that whole business where it makes the virus inert, where it just holds the thing together because the two proteins that need to come apart to let the virus let its genome out, it just can't do it, So there is another way besides just the potent neutralization. Although if I have a particular virus, yes, I absolutely want the super potently neutralizing antibody if I can get it, like no question.
How do you then identify these, the best antibodies? So you mentioned that you have, in some cases, samples before and after an infection. So that's a distribution of various antibodies. So how do you identify the one that to use as a benchmark or to go forward with for therapeutic purposes?
That's the hard part.
And that's the key question, right?
It's very complicated. I don't know, Matt would probably agree, but I think one thing about antibody discovery, like most science, I would say, it's a humbling journey. The more you do it, the more humble you become as you realize that far less than you wish you had. So one way I like to think about it is don't overthink. Yes. So overthinking would be, I think I know what I'm looking for, and therefore I'm going to define a tool set that gets me that thing. And then you do find what you're looking for often, but it actually turns out not to be really that interesting, right? What you thought was the solution was actually not necessarily the solution. So all that to say is, I think what we try to do, I'm sure Matt tries to do, is look at the virus. We obviously want function. So I guess if you put together neutralization and Fc effector, ultimately you want function, antiviral function of some kind. And what we like to do is think of how many different ways could we measure antiviral function? And let's put all of those in play. And some things like, can we just do simple things like say, can the antibodies actually bind with sufficient sort of binding strength to the target? That can be a very misleading thing, honestly, but in the context of function, it becomes an interesting factor. So really, I think what you try to do is put together a comprehensive strategy that admits that you don't actually totally know what you're looking for. But if you put those together, what you do is also a Pasteur quote, chance favors the prepared mind. So what you do is prepare as much as you can, and then you can call it serendipity or chance, but really it's because you set up like a sophisticated network of assays, not knowing exactly what was going to work. And then often it just emerges, this is what the best ones look like. They all fit this kind of profile. We never would have known that had we not had this comprehensive strategy for testing. So,
it's funny you bring that up. Part of the ReVAMPP goals is to try to also not just have these, it's actually 7 different centers, but not to have these 7 centers, but also to try to cross-pollinate with them. So I was at a meeting with, Jason McClellan, who's this structural guy who had, done all the coronavirus work in advance. And I said, hey man, will you come to my poster? I really want to see your thoughts on this thing. And I was showing him this story that I've been hinting at where, we have this like these 2 monoclonals, they hit this one epitope. It's like really the gold standard epitope for this particular virus, enterovirus D68. How do I apply that to all the other picornaviruses? That was kind of the idea. And, he just sat there and he kind of like looked at what we were talking about. And, I'm like, how can I bias my screen this way or that way? And he just said, he's like, I don't know, man, there is just something nice about casting a broad net and seeing what you get. Like, and like, he's like, you already found this without biasing your screens. So like, why would you change it? Like, just keep casting that broad net and then, see what comes out of it. And so, that's another person who I think we would all say his opinion carries some weight and just, he thinks the same way. It is nice to not bias too much, but also in a pandemic where you are trying to get the fastest route to the fastest thing, if we can, again, make a playbook where we at least have some thought that we might be able to target the receptor binding domain of SARS-CoV-2, and we actually know what that would be because we know what the RBD looks like from SARS-CoV-1 and MERS-CoV and all these other things. Well, you might try both an unbiased broad net approach and let's also like really look at that RBD. And
Absolutely.
So kind of the same thing. So, Jason did also give me a nice idea about how to try to maybe find those, neutralizing antibodies for other picornas. But, I really took it to heart when he was like, I don't know, man, like, why would you? Like, just keep going broad, just keep looking for everything. So like Rob said, well, let's just maybe do all of it, at least right now in our sort of information gathering phase. And then if we keep finding the same thing, even with our broad approach, well, then we've got a pretty good idea. Well, even with the broad approach, we are always still kind of finding the same thing. So that is really the RBD equivalent. Like we really do want to go after this particular site.
Yeah, I think it's very good to be, taking an agnostic approach. But then when you're repetitively finding the same thing, or it's an immunodominant site, but you want to find something that's very rare. And sometimes those are the most interesting and most potently neutralizing antibodies, using some kind of focusing, like epitope focusing, either, blocking with an antibody or glycan masking. Those are other ways you can apply approaches to find those antibodies. And sometimes you might even find the same thing, but it's a stronger affinity or neutralization, higher neutralization potency. So it's just, casting a broad net initially, because honestly, we don't know what we're looking for until we find it. And then, going backwards on trying to figure out how things function. And then, down selecting from that information to find rarer populations that might, be more beneficial.
And I think a corollary to what we're all saying here is it really is all about your screening process. So it used to be kind of tricky to be able to find human monoclonals. It's not really tricky anymore. That part's fairly straightforward these days. And so anybody can do it. So why should we be calling ourselves the experts who are doing it? Well, I think we're we've spent a lot of time, the three of us and the group that we've been part of, a lot of time trying to think about what are sort of the best ways to look, because as Rob said, if you screen for something specific, you're likely to find the thing that's specific. But if you've outsmarted yourself, well, then you've outsmarted yourself. And so how do we avoid outsmarting ourselves or using a screen that's unintentionally, biased towards something? And, I spend a lot of time thinking about that versus other things, because if we're going to launch a discovery effort, I want it to be worth our time and I want it to be worth our effort. And so that's where, again, taking a couple different approaches might help you avoid that bias too. But the screen is everything. It's everything.
But ultimately, you need a sequence, right, of the antibody. So how do you, if I understand correctly, you start with serum samples to identify the potent ones, right? No?
Boy, you can see us all like screwing our faces up, because that is also a different, super complex question. I feel like I've been talking too much, so I feel like I shouldn't take this one. But Lauren. Lauren, it's your turn.
Yeah, sure. Instead of serum samples, taking PBMCs, so peripheral blood mononuclear cells, and isolating B cells from those PBMCs, that's usually, that's where we're getting our antibody sequence from. Serum proteomics and analysis, that is another way of doing figuring out the antibody sequence. But for the three of us, that's our approach, is looking at PBMCs and the B cells isolated from there, and sequencing B cells of interest, whether that's a pool or even at the single cell level, and using sometimes antigen to isolate specific B cells, whereas other times we're being a little bit more broader in that sense of memory B cells, or even, plasmablasts are other ways that we're, isolating antibody sequences.
I'll just add in there, one of the key parts I think built into what Lauren's saying is you can do serum proteomics, or even, sequencing, sort of large scale sequencing of the antibodies in one's circulating antibody repertoire. And that's all interesting in its own right. But what we have to be able to do for therapeutic antibodies is connect antibody sequence to antibody function, right? So we have to predicate all our methods on having literally individual B cells in our hands that we can say, I have assigned a function to the antibody being made by this B cell, and therefore the sequence that I obtained from it, I can logically connect the dots between this sequence and that function. So when you're doing larger scale things, you can understand polyclonal immunity and sort of community and global aspects of immunity. But therapeutic antibody developments, you have to kind of somewhat let go of those concepts and let other people study those and narrow in to hardwiring the connection between that sequence and a function that you're looking for.
I was going to try to tie this back to, since this is a adaptive immune receptor repertoire podcast. Curious, obviously, it's sort of, you said earlier, the ReVAMPP, one of the mandates was kind of learnings, right, from the past, from the COVID pandemic. And so your current platform that you've been talking about is focused on pulling, kind of using infected individuals as kind of not a hybridoma approach, but just as a model of generating a bunch of antigen-specific antibodies, and you're pulling out the functional antibodies. I think in the past you've mentioned, right, that they're probably like the memory B cell phenotype, et cetera. But I'm curious in terms of on this population-based level, particularly with the coronavirus, where there was, there's now kind of a longitudinal aspect, and then also with all of the allelic diversity in terms of the BCR, but then also just the other alleles and genetic differences that people have that could have different repertoires. Are there efforts to kind of understand retrospectively, right? Or because obviously there's people who get the vaccine who might not, be able to generate those protective antibodies against those certain epitopes. Are there people looking at that aspect?
I would think on the vaccine side, yes. We can fortunately sidestep all that because we can program in to the antibody, therapeutic antibody, essentially like targeted function and functioning in both the FC side and the FAB side by just how we build the molecule. But I get what you're saying. On the vaccine side, it's very important for it to understand, what kinds of antibodies would be protective, as Matt was, and I think Matt and Lauren both talked about earlier. And then how would you elicit those, and what is the background into which you're eliciting those, i.e. the humans? What's the interface of the human, genetics of the human and your ability to elicit? If that's what you're asking, yes, people are studying that. We're not those people, but people are studying that. I would say the repertoire part for us, if we want to connect up to repertoire just a little bit, we did talk about very narrow discovery things, which might imply that we don't actually look at like larger sort of antibody collections and repertoires. That's not completely true. We actually do. What we do often is do very narrow focused studies to help us understand, here are some examples of neutralizing antibodies. Here's the kinds of things they do. Matt was mentioning a couple. Here's kind of how they work. We can in parallel process larger batches of B cells where we do get this non-functionally connected set of antibody sequences, thousands, tens of thousands of these antibodies. To us, we can still use that for therapeutic discovery once we have that information from the specific approach. Now we know what neutralizing antibodies look like. We understand something about how they look. Then we can use that information to mine these larger sets that we've gotten through sort of non-targeted or less targeted approach where we're just looking at, for example, blasting B cells in an acutely infected person, or in memory B cells that have been just sorted for memory B cell markers, or even antigen-specific ones, but it's a really large set of sequences that are antigen-specific. We can now start to make guesses as to, I think this group here looks like it would have a function similar to these ones we discovered based on how they look. And so we can use both sides of it as long as we can do the first part well.
Yeah, I think as part of the revamp, it's not an active part of the effort. It's more to find antibodies and, more on the vaccine side, as Rob was mentioning, but we're still getting the large data sets of antibody repertoires, and then being able to look back, from the specific to, antibody siblings, but then we're also having multiple individuals that we're getting PBMCs from that we're able to then look for, are there similar, shared sequences between these people? And even, just from other studies, not even necessarily from the revamp, that we have these sequences, can we look and see, maybe this person was asymptomatic, but they have this antibody, and is that why they were asymptomatic? Because they had these great neutralizing antibodies, or, antibodies with FC effector functions that, provide protection. So, that information is there. It's not just necessarily, exactly what we're looking for. We're trying to better understand immune correlates of protection from the, and also having the antibody as a therapy.
And I believe there's a ReVAMPP group, maybe Lauren and Matt can correct me if I'm wrong, based out of the University of Washington, which is more computationally focused. So they can even take the information, like we just said, we would generate these large antibody repertoires, take some of the information that they're generating, and then basically train learning computational systems to make antibodies from scratch, or change and alter and improve antibodies that have been discovered through traditional means. So these might also be usages of these large data sets to train people who are doing more computationally oriented projects.
And while it's not our project, but it is something that fits with like trying to make the connection to the function and the repertoire, so there's an effort in HIV, and I happen to be, right up the hall from Christina de Paris. So hers is a lab that's like 2 labs down from me. Nothing to do with ReVAMPP, but she's an HIV researcher, and they have kind of taken this approach where they know, HIV has had lots and lots and lots of antibody discovery work over the course of time. And as Lauren has pointed out, that's one of those cases where like there are rare like epitopes that antibodies are rarely made against, but have been recognized to be these like broadly neutralizing epitopes. So, hey, that's really cool. And so they've kind of tried to reverse engineer where like they know what the end goal is. And so how do you vaccinate someone to get them to make that antibody? And what they're doing these days is like sort of trying to change the vaccine antigen as they, give their first one and then boost. The vaccine antigen changes to try to basically direct the repertoire and really focus it towards that antibody, that epitope where, the theoretically in general, we don't make a lot of antibodies to, but can we figure that out? And so as I understand it, a lot of that work again came from reverse engineering. So you have the sequence and you can sequence. I guess another thing to think about is the difference between sequencing a bunch of plasmablasts shortly after some immunogen happens or infection happens versus memory B cells, some period of time after that happens. But the idea is like trying to watch either whether it's you can predict it or whether you can actually watch it, depending on how your study is set up and the samples you collect from humans. But watch the antibody go from the germline, VDJ, whatever that is, whatever those 3 genes are, and then actually somatically hypermutate along the way to becoming this, giant monster of an antibody with like a super long CDR3 that, really, buries itself into a particular epitope or something like that. And so that is definitely one way that I know of that, a whole field of people has been working on trying to combine repertoire studies that are very repertoire studies, but also with functional studies. And I think that's what Rob's getting at and Lauren are getting at as well when they talk about like CurlLab has a lot of repertoire information from studies that were more like the one I described where you're trying to watch the evolution over time of someone's antibody repertoire. But then if you have a function for a particular antibody, you can kind of go back to those data sets and see if you can find them a lot. You can see if you can find them at different stages of development. And then at least theoretically, can you design a sequential vaccine strategy that takes a broad antibody response to start and then focuses it really more towards getting the B cells you want that are making the thing that you want. That's beyond my level of like abilities, but that is a way for someone to take an immune repertoire and use the functional stuff and try to guide it.
With the goal of developing the therapeutic antibodies, is it the ones that you're proposing, are they the fully endogenous ones that you're pulling from people, or are they further engineered for higher affinity or function or effector function within the FC, et cetera?
So I'd say for the most part, we keep, the endogenous sequence. And regarding changing the FC, modifications, usually we're modifying to increase half-life. So from a therapeutic antibody standpoint, you're having longer lasting immunity. But we are working more and more as we're, AI and machine learning is growing, especially in the antibody space, and even modeling of antibody antigen complexes with like AlphaFold 3, for example, being able to predict more when a structure isn't like high resolution structure isn't always available, being able to computationally redesign antibodies as well. But for the most part, we are still leaning towards the endogenous. It's still kind of earlier stages, but it's the more and more we're doing it, the more these models are becoming better and better at being able to do so. So I could foresee in the future that it's more common to computationally redesign.
Yeah, it's one thing to think about improving affinity, but you're walking a fine line. You're also potentially increasing the chance of autoreactivity, right? You're taking something which was a human protein and turning it into a not so human protein, maybe. And so that's where I think the computation for improving affinity is there. It's there, right? People can do this with what we're still, I think, now coming along and people are doing is now using actually repertoire data, right? With what you combine that with repertoire data, you can understand, well, at this particular amino acid position, what would a human tolerate? We know that from repertoire studies, right? In this position of the heavy chain, what do we ever see anything there other than these 3 amino acids? If not, then we better use one of these 3 amino acids because the humans is going to see this as not native, not normal. And so those things are merging. And I think we will get to a point where we can not only do computational retargeting or improvement, but de novo antibody discovery is going to happen. I don't know that it will happen and work in every situation because it's really dependent on the data sets that are available. But there will definitely be minimally discrete situations where people will do completely computational discovery programs and it will lead to something useful.
My last question was, what virus keeps you up at night?
That's a good one.
That is
all of them, to be honest.
I'll go with Rift Valley.
yeah.
An obscure one, Rift Valley fever virus. Not that it's widespread. I think it's mostly localized to Kenya. But the truly terrifying part of that is it's a pretty promiscuous infectious virus, and that it can infect humans and, importantly, animals that we eat. So not just random animals, but animals that we actively keep close to us, livestock, and sort of people are often in close proximity. So there, I mean, it's not happened yet, and it has some reasons it may not spread. But if it did somehow get in somewhere, you could imagine it being you're either infecting humans or you're culling entire herds of human, sort of food.
I'm in my head, I keep jumping between coronaviruses and flu and like filoviruses. I think those are the ones that I'm like bopping through. The filoviruses for kind of the same thing Rob said, minus the whole infecting our food, but like they're super contagious as long as you're like immediately in the vicinity of someone who has one of those infections. If they ever gain any ability to spread by anything other than close personal contact, that's really frightening, like terribly frightening. And even with, a lot of the mortality rates we think of with those are like, in the 30 to 50% range, but that's usually based in like an African setting. I think when you get filovirus infected people back into sort of like US facilities, the mortality drops to a mere 10%. 10% mortality is still pretty robust, and would cause a massive problem. Not to mention, like, even if you live, we know people who have survived these infections and have met them and spoken with them. I mean, they have sometimes debilitating lifelong sequelae, even if you, again, you survive and move along. So maybe I'll go with those just because I already spent some time talking about them. But don't get me wrong, I the others too. Yeah.
Yeah, if I had to pick one, it would be rhinoviruses, probably, because they're very difficult. They're very diverse. There's over 160 serotypes. So there's a lot of them. Everybody gets a cold, multiple colds over your lifetime. So that's probably what keeps me up at night.
Well, you've unlocked new fears for myself too.
Yeah. I'm not sure how to, on this happy note.
I know.
Hey, that was your question, guys.
Yeah.
Yeah.
That is a good one. Well,
I know, but okay.
So this brings us to the end of the 19th episode of onAIRR, the podcast of the AIRR community with a special focus on clinical use of the adaptive immune receptor repertoires. The podcast is supported by the Antibody Society. Please go to the website antibodysociety.org to get more information about our sponsors. If you have any comments, questions, drop us a line at onair@aircommunity.org or just tweet or X using the hashtag onAIRR with 2 R's.
Well, thank you so much, Robert, Matt, and Lauren, for speaking with us.
Thanks for having us.
Yep, thanks.
We are looking forward to inviting you guys all back for the next episode to hear some of your great stories of how you find these monoclonal antibodies in patients. All links and contact information are in the show notes. Thank you for listening to onAIRR.